Thank you very much. I'm sorry that for this poster I have no slides because Anu is ill and she sent me the data and the presentation for the poster, but in the email was only the poster in the PPT version, but no problem. The idea behind this protocol was to test the influence of exercise on immune cell mobilization and to find a method to measure it on a simple way. And we performed a 1-minute sit-to-stand test and measured on before the test and 1 day after. We included 18 ME/CFS patients concerning the Canadian consensus criteria, not distinguished between 14 hours PEM, more or less PEM, because we would see the whole range of the response. The Raman Spectroscopy is a method where light scattered to a molecule, and based on their stability of the molecule or elasticity of the molecule, you can measure changes in immune cells or molecules. And what we see is on the left side that patients and controls show responses concerning the exercise. We see an up-regulation for the patients and an upregulation for the controls. And if we plot this data for each patient, and there's a little bit too creepy to read it, but this means the visit 1 before the test, and this means the visit 2 after the test. You see there's a variation in response, in the immune cell response, and the mobilization of immune cells after the test. And we are at the moment happy with this test because the variation gives us the possibility to find and distinguish different groups, potentially different groups in response to exercise. But at the moment we will develop further the protocol of the anaerobic lactacid test, 1-minute sit-to-stand test, because the time points of measurements between day and 1 day after for this analysis could be not the best, and so we can develop it further. The study was funded by the ME/CFS Research Foundation, and thank you for this funding, and is the basis for the BioSig-PEM project, where we can develop this method further to distinguish, based on Raman spectroscopy, patient groups in immune cell mobilization, to get a better understanding about the interference of exercise-induced and infection-induced immune response. Thank you very much.
Thank you, Christian.